Rabies is a viral disease which is transmitted to all mammals by several vector species, essentially carnivores, including dogs, but also bats, for example. This is a neurotropic virus, meaning that the virus has to travel from the place of inoculation to the brain by using our neurons. This corresponds to the incubation period, which can take time, 2 months in average. It is followed by the symptomatic period, which is short and which systematically links to death. Thanks to vaccines, rabies is a preventable disease which can be prevented both in animals and in humans. Vaccination of dogs, for example, in the context of One Health approach, is very positive to protect humans against rabies. Unfortunately, in many developing countries, vaccines are not available in sufficient quantity to protect population. On the other hand, in developed countries, some people do not have recognition of rabies and are not going to be vaccinated, for example, after a bat exposure. For that reason, rabies is still killing 55,000 people every year in the world. Rabies is a neurotropic disease, and rabies is provoking modification, comportamental modification in animals. But these modifications are not constant. In dogs, for example, frequently rabies makes dogs furious, but not systematically. So clinical diagnosis cannot be 100% appropriate. For that reason, laboratory diagnosis is needed. Laboratory diagnosis is done by euthanasia of the animal and look in the brain if rabies antigens are present. The gold standard methodology is immunofluorescence. In addition to that, other methodology exist, sequencing, for example, that allow to know the source of the infection and to make molecular epidemiology of rabies. We have to know that vaccine against rabies exists since the time of Louis Pasteur, more than 100 years ago. These vaccines were produced on nervous tissues with several side effects. Since decades, cell culture vaccines are available, they are safe and very efficient, and then they can be given both to humans and animals. The problem is that you have to catch the animal to give these vaccines in order to inject them, and this is not all the time possible. For that reason, oral vaccination has been developed essentially in wildlife, and it was a big success, for example, in Europe, to vaccinate foxes through baits. We can imagine that similar development could be done in the future for other carnivore species like skunk, or even for dogs in developing countries where they are not systematically available. I'm talking about the wandering dog. This is the first scientific approach for the future. The second one is that since decades, we start to know now that there is one disease, which is rabies, and many viruses that are Lissa viruses. And these viruses are not similar each other. And so, in the future, it would be nice to develop anti-rabies vaccine and to transform them into anti-Lissa virus vaccine, meaning to increase the spectrum of protection of the vaccines to all Lissa viruses. And this has to be done through different types of recombinant technology with molecular biology.